Hi everyone. In this video, we're going to be talking about adaptive immunity. And adaptive immunity has a whole bunch of synonyms, including specific immunity, specific resistance, and acquired immunity. So any of those are okay alternatives if you're describing the adaptive immunity. And basically, when we talk about this, we're talking about the body building specific defenses to very specific exposures in order to protect the body from various types of infection and disease. And so we can actually divide and sort of classify this immunity into a couple of different categories. And I think it's just useful to do this to help us think of the common examples that the immune system is going to interact with, but certainly some will be much more prevalent than others. So first we can divide acquired immunity into two categories, active immunity and passive immunity. And active immunity is produced by exposure to antigens. So think of an antigen as something that is possibly dangerous to the body. And in order for active immunity to be functional, there has to be an actual exposure to an actual antigen. Conversely, passive immunity is produced by transfer of antibodies from another source. So it doesn't have to be exposure to the antigen, just administration of the antibodies in some form or another. We can further break down these classifications into four basic categories. And these four basic categories, I think, are definitely the things to remember because when we consider the examples for each, I think it's easier to sort of keep in context what the immune system is doing in these different circumstances. So first we have naturally acquired active immunity. Second, artificially induced active immunity. Third, naturally acquired passive immunity. And fourth, artificially induced passive immunity. So notice that we're using the word active or passive from the previous category in this classification. And then we're adding either naturally acquired or artificially induced to it. And I think it's definitely okay to just say natural active or induced active or natural passive. But I used all these words because different textbooks use different terminology, and I just want all the words to be familiar. But you could shorten it up a little bit as long as you are including whether it's natural or induced and whether it's active or passive. So naturally acquired active immunity is probably the most common situation, on a day-to-day basis that the body needs to sort of utilize and make function. And this is commonly resulting from exposure in the environment. So if if there's a cold virus floating around or a GI bug and you touch a door handle and then you touch your face and next thing you know you've taken in somehow that bug. Now your body has actual exposure to the antigen and it needs to develop a response to get rid of it. So the type of immunity that it's going to utilize or develop is the naturally acquired active immunity. And I really want to sort of just specify that active immunity means that the body actually has to do work in order to develop the immunity. So upon exposure to the antigen, it's gonna physically make some antibodies. It actually has to do several processes in order to develop the immunity. Next is artificially induced active immunity, and this is basically administration of an antigen on purpose. So commonly this would be a vaccine. So if you think of a vaccine, generally it's an injection that's given, there are obviously different types of vaccines, but thinking about, children and they have scheduled vaccines, every so many months or every so many years, depending on how old the child is, for things like measles, mumps, rubella. Now they have Varicella Vaccine, which is chickenpox. So all sorts of vaccines where they basically give you either like a dead virus, so sort of like, maybe protein components of the virus or just something that's not live, or if it is live, it would be attenuated, meaning that it's severely weakened. So you're giving the body just like a sort of a minimal exposure to the antigen and then asking the body to develop the immunity to that antigen so that upon subsequent exposure, the body won't get sick. The body will have adequate stores of immunity to deal with the particular antigen. All right, next we have naturally acquired passive immunity. And so this is the example here would be thinking about like if you have a pregnant female who's carrying a fetus, she will transfer antibodies from her blood to the fetus So generally we think of this as transfer from an immune individual to a non-immune individual. So mom to baby is the easiest and most simple example here. And so basically the antibodies that mom has built up throughout life can be just sort of can cross the placental barrier and just sort of protect the fetus, especially in the first couple of months of life. And another example would be transfer of those similar antibodies in breast milk. So if mom is breastfeeding, then that would be another means by which the fetus or at that point, the newborn would be able to acquire those antibodies. So the thing to know about passive is that because those antibodies are coming already ready as opposed to coming in the form of an antigen that needs antibody production, there is no work that has to be done, which is why it's considered passive. And the final example is artificially induced passive immunity. So this again is going to be likely like the form of an injection, and we could think of administration of something like antibodies through an IV, like certain patients who would get like IVIG as an example. So that's also passive in the sense that the antibodies are just being administered. And in this case, it's important to note that these antibodies that are being administered are not going to become part of the body's continuous immunity, so they can help assist in fighting a particular infection for a short period of time while they're being administered, but over the long term, the body doesn't develop immunity to whatever that particular antigen was that was causing the need for the IVIG. So definitely keep in mind that is temporary, as opposed to the active forms, which are permanent. They can develop the immunity for the long term. All right, now I know we've already talked about the non-specific defenses in terms of how they can sort of prevent or quickly mitigate any type of exposure to an antigen or a pathogen. But when we think of the immune response in the context of adaptive immunity, I want to just sort of put into perspective some of the things that are going to happen so that we can consider the different processes very, very carefully. So think of a virus or a bacteria, so some type of pathogen that is going to enter the body. And so this person obviously is going to be feeling pretty sick and pretty unhappy. And then the one of the things that needs to happen right away or early on is this process called antigen recognition. And antigen recognition in turn will confer an immune response. And we haven't really gotten to like what the specifics of that immune response are yet, but I just want to make a couple of statements about antigen recognition and about sort of the process that goes along with it. So an antigen is a pathogen, part of a pathogen, some type of foreign substance. It's usually protein in nature. It could be polysaccharide. Very rarely is it lipid or nucleic acid or something else. Usually protein, and secondary maybe a sugar of some type. Now, antigen generally is considered a bad thing in the body, but it's also important to note that the body produces some of its own antigens, and so its own antigens would not be dangerous to the body, but would be dangerous if they were injected or inserted into someone else, like another individual, in which case they would be classified as bad. So for the purpose of clarity for our discussion, we'll classify antigens in one of two categories. We'll either call them foreign antigens, in which case we think of them as bad and in need of an immune response, and then we'll classify the others as self-antigens, things that the body naturally produces and sort of displays to different types of immune cells. So I will always be specific and say foreign or self in order to make sure that we know sort of what the expected outcome is going to be in response to the exposure. So we have this idea called antigenic determinants. So antigens have specific sites on them that can be recognized by the immune system, and those sites are called the antigenic determinants. And so not every piece of the antigen is necessarily bad or is necessarily visible, but those particular sites can be recognized by the immune system. And so I want to introduce this idea of specificity. Let's just draw a couple of different antigens, and you can see that they're antigenic determinants. So these little sites that can be recognized by the immune system are different. So we have just these little spikes, and then we have these more like triangle spikes, and then we can have different immune cells sort of come over, and the most scientific way of describing it is that the receptor site would match sort of the shape of the antigenic determinant. And so there are different receptor sites on the different types of immune cells that can match up with the different antigens. And so specific immune cells will recognize specific antigens. So A recognizes A, B recognizes B, and they can only target the specific antigens that they're able to recognize. So if you think of an immune cell only being able to kill one very specific type of antigen, but then you think of all of the, just like thousands or millions of types of bacteria and viruses and other types of pathogens that exist in the world, the body would need to be able to mitigate most of those. And so we have this other idea called versatility, which basically means that there are so many lymphocytes, probably in the trillions, that are generated to deal with the different types of antigens. So we just make enough to deal with the different types and still maintain specificity. Another property that's important to note is tolerance, which is also self-tolerance, depending on what book you're reading. So basically, if the antigen is displaying an antigenic determinant that is self, then the immune system has to recognize that antigen as self and not destroy it. So that's important because when we think about the body destroying its own cells, that's how we classify an autoimmune disease. We start destroying our own cells. We start to develop disease as a result of it. So we can't have the immune system be broken. We want to make sure that it can recognize the things that belong to it. All right. So when we think of immune response, there are two sort of processes involved in developing the immune response. One is cell-mediated immunity. And in cell-mediated immunity, we can think of, so let's just draw a body cell. And in the body cell, we can see nucleus, endoplasmic reticulum, Golgi apparatus, and a virus. And so the virus has been sort of engulfed, or it's made its way into the cell. And it's actually, it could be classified as hiding in there. So if cells just sort of float on by, you might not be able to see it, but the body's going to have an interesting process that we're going to discuss shortly to make sure that especially the immune cells that are floating by are able to see these infected cells. The second type is humoral immunity, and humoral immunity is also called antibody-mediated immunity. And in this case, we could think of the antigen or the pathogen sort of just floating in body fluids. And there are obviously different types, but maybe it's like a bacterial cell or something. And then we have our immune cells. And in this case, the pathogens will be visible. And so there can be various steps to attack those pathogens as well. All right, so before we can talk about both cell and humoral immunity, we need to talk about one other idea, which is the major histocompatibility complex. And it's MHC for short, and alternatively referred to in humans as the human leukocyte antigens, or HLA. So I will exclusively say MHC, but in case there's maybe another reference or source that refers to it as HLA, I just want you to know that it's the same thing. So basically, the MHC is a set of genes that codes for proteins. And so these genes are genetically inherited, one from mom, one from dad. And they make basically two classes of MHC proteins. Class 1 MHC proteins are found in all nucleated cells. So every cell that has a nucleus, which, the things that we've mentioned, for example, like red blood cells don't have a nucleus, so we wouldn't find class 1 MHC proteins on those cells, but any cell that has a nucleus, we definitely would. And the second is class 2 MHC proteins, which are found on these cells called antigen presenting cells, or APCs. So we haven't mentioned APCs before, but think of them as just sort of an umbrella term to describe cells such as macrophages, B cells, and dendritic cells. And mostly when we think about those cells, what we really want to think about is what the importance of the immune function of those cells is relative to the MHC. And we'll get to that shortly. All right, so let's consider the mechanism for class 1 MHC. So this is a body cell. This could be any body cell. Think of it as as long as we think of it as a nucleated cell, we're good to go. So we can see the nucleus, endoplasmic reticulum, and Golgi apparatus inside of this cell. And under normal circumstances, body cells are going to degrade things like endogenous proteins. So if you think of like a membrane receptor that has is old and damaged, the cell is going to degrade it. So there's a specific set of proteins that will be degraded by the cell that could be used to make sure that immune cells know that this is a body cell. So as the protein is broken down, it'll be taken into the endoplasmic reticulum and incorporated into the newly synthesized class 1 MHC protein. And then once the synthesis is complete, it'll be inserted into the membrane. So we can see class 1 MHC protein is now sitting in the membrane, and it's holding on to a fragment of this degraded protein. And so this is a self-antigen. So this, when the T cell floats by, and we haven't mentioned T cells specifically other than that they're a type of lymphocyte, but T cells are going to be the ones that are mostly going to interact with these proteins, so those are the ones that we'll draw. So T cells are going to recognize this antigen fragment as self, and as a result of that, they will just float on by. However, if instead of displaying an antigen fragment that is self, this particular cell has been infected, and that's very possible. It could have been infected by virus or other type of pathogen. If this cell's been infected, then instead of it being an endogenous protein that was degraded and then presented, it would actually be a pathogenic fragment. So we'll just say foreign antigen, and if the foreign antigen is instead displayed, then the T cell is going to recognize that, and then what will result is an immune response. So we'll see the class 1 MHC again, and we'll talk when we talk about the specific role of the T cells. We'll see that we'll see kind of the specifics of this. All right, so now let's draw another cell. This time we're going to talk about the mechanism for class 2 MHC, and we're going to draw an APC, or an antigen presenting cell. So let's pretend this is a macrophage, and the macrophage encounters a bacteria. So this is a bacterial cell, and the macrophage is going to engulf the bacterial cell by phagocytosis. And let's not forget that these cells are going to still have the nucleus and the endoplasmic reticulum and that sort of thing. And after the bacterial cell has been engulfed, we can actually see that it's sort of sitting in a vesicle inside of the cell. And a lot of times these guys will fuse with things like lysosomes, and then the bad thing that's been ingested will be degraded or fragmented. And then in this case, what we're also going to do, so we're going to be producing the class 2 MHC in the endoplasmic reticulum, similar to the class 1. Once it's synthesized, it will be released in the form of a vesicle, and it will fuse with the vesicle that contains the bacterial cell or the bacterial fragments. And then in that new sort of formed vesicle, a foreign antigen fragment will be inserted into the binding site for the class 2 MHC. And then the class 2 MHC will be taken to the membrane, and then we'll have a display of the antigen fragment that is foreign. Now in this case, if we have a T cell or immune cell that comes by, it will recognize the foreign antigen and then generate an immune response. So it's important to note that the immune response that's going to follow the class 1 versus the class 2 recognition is going to be completely different, but there's going to be some overlap with the different types of cells, and it's just useful to sort of keep these mechanisms in mind in order to make sense of the functions that follow. All right, so that wraps up our discussion and overview of adaptive immunity, and moving forward, we'll be talking about T cells and cellular and humoral immunity, as well as the functions of antibodies. So I hope you enjoyed the overview. Thanks for watching.